In this video, I will be discussing about the Griffiths experiment, McCarty and MacLeod experiment, and Hershey and Chase experiment. So Griffiths worked with two strains of Streptococcus pneumoniae, a virulent disease-causing strain and a non-virulent strain. He observed that when he injected mice with live virulent bacteria, they died, but mice injected with live non-virulent bacteria survived. Here you can see this rough strain. When he injected into the mouse lives because it is non-virulent. But the smooth strains are virulent, and when Griffith injected this into the mouse dies. Then what he did, he heated the smooth strain and then injected into the mouse. Then he was surprised to see that mouse lives. So then he mixed the rough strain and heat killed smooth strain and then injected into the mice. Then still the mouse dies. So Griffith, this suggested that something in the heat killed virulent bacteria transform the non-virulent bacteria into a virulent form. This phenomenon, later known as the transformation. So after some years, like in the Griffith experiments were done in 1928, then in 1944 McCarty and MacLeod wanted to identify the transforming substance which is responsible for this bacterial transformation. So they treated the heat killed virulent bacteria with enzymes that specially degraded proteins, RNA, or DNA. So enzymatic destruction of DNA prevented the transforming ability, while the destruction of proteins or RNA did not. This indicated that the DNA was the genetic material responsible for the transformation observed by the Griffith. Here you can see when he added the proteinase which cleaves the proteins, then here you can see the S cells appear. When he added the RNAs which degrade the RNA, still the S cell appear. But when he added the DNAs, that the substance which degrade the DNA, then he was surprised to see that there were no S cells appeared. So the conclusion that transformation requires DNA. Therefore, it is a genetic material of the cell. So McCarty and MacLeod confirmed what is that substance which is responsible. So here you can see they proved that DNA is the genetic material of the cell. Then later in 1952, Hershey and Chase used the T2 bacteriophage, a virus that infects bacteria. T2 bacteriophage. They radioactively labeled the viral DNA with phosphorus 32 and the protein coat with sulfur 35. The labeled T2 phage were allowed to infect in E. coli bacteria. After infection, the viral protein coat remained outside the bacteria. Only the labeled DNA entered the bacterial cells. This confirmed that DNA, not protein, is the genetic material that carries the instruction for viral replication. So later Hershey-chase experiment 100% proved that DNA is the genetic material. So, what's the conclusion? Is that the Griffith experiments demonstrated the bacterial transformation. Then McCarty and MacLeod identified DNA as the transforming substance. Later, Hershey and Chase confirmed DNA as genetic material using a viral system. Collectively, these experiments established DNA as the molecule responsible for carrying the genetic information. They laid the groundwork for the understanding of the molecular genetics and the pivotal role of DNA in heredity and biological processes. Thank you.